Okay, so yeah, now you hear me. Good. Okay, so did you hear before? So yeah, today we have two former students of the degree from the same promotion. The second one, they finished both in 2020. So the first one to present today will be Marta Ibáñez. Marta Ibáñez, I was telling that she did the final project in an internship in Cambridge on diseases related to pregnancy, and she took 2 masters, one in genetics and genomics, and one in translational biomedical research, and she has just started a PhD in Vall in BHIR just very recently. Okay, so her presentation will be today, my journey from bioinformatics to the PhD. Thank you. Thank you very much, Marta.
Thank you. Well, hi everyone. As Arnau has said, my name is Marta, and I'm a first-year PhD student. And today I thought that I could walk you through from the beginning, when I was just in your place, till now, where I have started a PhD. So actually, my journey starts in 2017. When I applied to bioinformatics. So in fact, I didn't know that I wanted to study bioinformatics. I knew that I wanted to do something related to health science, but I wasn't sure what that was. So one day, my mom was reading the newspaper, and she saw this interview where someone, who we don't know who it is, was saying that bioinformatics was going to be key for health science. So that's when I decided that it looked appealing, so that I could do it. So around September 2017, I started this bachelor's degree. So now I thought that it would be a good idea to give you a bit of advice of how my experience was in this bachelor's degree. So from my first year, actually, I struggled a bit, specifically in my first trimester. I think now it's a bit different, but in my first trimester there was calculus, and it was a horrible subject. I didn't like it at all, and I had to go to recovery exam, but I passed in the end, so everything was good. And then I liked programming, but at the same time, it was very difficult for me to get in this programming mindset. So I struggled a bit, but I also passed, so there was no problem there. On the other hand, there were also subjects that I liked, such as algebra or genetics and genomics. So in my second year, I think I liked it a bit more. However, there were also some subjects that were not my favorite ones. And for example, there was structural bioinformatics, which you will see it's quite complex, and it has a lot of content, but you can overcome it, so don't worry. And then there was also programming and algorithms too, which it's not as hard as I had it in my mind, but I think it was because I had this very closed mindset in which I thought that I had struggled a lot during my first year. But in the end, it was fine, and I didn't have to go to recovery exam or whatever. And then other subjects that I liked were databases and omics techniques, specifically because omics techniques opened my mind to the field in bioinformatics I am currently working on, so meaning sequencing and all of these things. So I was really excited for this subject. And then during that same summer, I did an extracurricular internship in the synthetic biology for biomedical applications lab at Universitat Pompeu Fabra, in which I could apply my programming skills and a bit of wet lab. At the same time, I need to remind you all that during your second year, or at least that's what you should do, is basically contact potential labs in which you can do your final grade project and curricular internship. So I struggled a bit because I knew that I wanted to go abroad, and I had to send a lot of emails and do a couple of interviews, but it's all worth it, I promise. And then finally, my favorite year, although there was COVID-19, is my third year, where I was able to do my final grade project in the Ferrucci lab at the University of Cambridge. However, I want to highlight a couple of subjects that are not these ones that I also found pretty interesting, such as data visualization, which I thought that it was really enjoyable, and you learn a lot about graphs and everything. And then also, it's not in the list, but computational genetics and genomics is pretty important, and at that time, it's really hard, and you will have a lot of work, but it will be worth it if you want to work in sequencing, because the things that I learned at that time are things that I'm using now in my day-to-day life in my PhD. To go a bit further into what I did in my final grade project, well, I joined the Sferrucci lab, which is run by Professor Amanda Sferrucci, and her interest lies in studying pregnancy complications from the placenta's point of view. So to give you a brief summary of what I did, I basically joined this project whose aim was to identify biomarkers from the placenta that were secreted to the mother's bloodstream in order to detect pregnancy complications in women. So how this worked was that there was basically mice at day 16 of pregnancy who, of course, were pregnant and had around 8 to 12 placentas. So these placentas were extracted and cultured, and then we performed mass spectrometry, which that means basically that you are detecting the peptides that are being secreted in your culture. And finally, that's when I joined doing the peptidomic analysis, in which basically we gathered these secreted proteins and compared them to a public database that you may know that is called UniProt, and looking at its corresponding genes and seeing if these were actually related, had been seen to be related to other pregnancy complications in published data. And so this was very exciting for me. However, due to COVID-19, I had to go home to end this project. But also, this gave me my first paper in communications biology that you have the link in case you want to read it and see the results. So once I finished my bachelor's degree, I had to ask myself what I wanted to do next. I was not sure if I wanted to do a PhD at that time, but I knew that I liked research, and my supervisors back in Cambridge had told me that I had potential to do research and pursue a PhD, and that I had a very good bioinformatics background. However, I lacked a bit of interpretation of results. So that's why I decided to do a master's degree in genetics and genomics at Universitat de Barcelona, to learn more how we could interpret these sequencing results in research. So this master's degree is basically based off 4 months of lectures, and then you have 6 months of just doing your master thesis. And there are 2 specialties, but the one that I chose was human genetics and genomics. So for my master thesis, I decided to rejoin the lab in Cambridge, and I did there my master thesis, which was based off the results that I had gathered during my final grade project. So basically, I had seen that there were genes that were differentially expressed in pregnancy complications compared to normal pregnancy. So we decided to select one of these diseases, which is gestational diabetes, and gather different data, which is transcriptomic data, which is basically human genetic genomics data regarding RNA sequencing, and DNA methylation data. That I don't know if you know what it is, but basically what leads alterations in gene expression is led by epigenetic marks, such as DNA methylation or RNA modifications. So to gather this information of DNA methylation data, I selected studies that had performed bisulfite sequencing to integrate this data and basically identify common potential changes in gene expression and methylation in placenta that could contribute to this pregnancy complication. As for the results that I got, I didn't find any correlation between differential expression and differential methylation, meaning that probably this type of epigenetic mark, which is DNA methylation, is not leading changes in expression in placenta in gestational diabetes. However, I did find common altered genes in this integration of data and common methylated genes. So again, I finished this master's, and I knew at that time that I wanted to pursue a PhD. So what I decided to do was go into another master's and apply for it, and this is the Master's of Translational Biomedical Research that is done at Vall Institute of Research and Universitat Autònoma de Barcelona. And while I was applying to this master's degree, I met my current boss, who is this guy with the arrow. His name is Dr. Josep Quer, and he runs the liver disease viral hepatitis lab back at Vall. And well, he saw my curriculum, and he was very impressed that I was a bioinformatician, so he decided to hire me and for me to do my master thesis in his lab. However, this lab was a bit different because, as I have mentioned, if you really think about it, I was really focused on human genomics. And that is basically was with the aim to understand the function and the structure of these human sequences. However, this is a virus lab, so I was focusing on infectious diseases. So how do you analyze infectious diseases? It's done a bit different, and it's done with a technique that is called metagenomics, which allows the detection, identification, and characterization of infectious agents. So this is a bit complicated, so I hope you can follow me. But basically, this is done with a wet lab technique that is called random priming. So before you sequence anything, this sample needs to go through a PCR that hopefully you know all of it because of COVID-19. And you need to amplify the genomes or the sequences that are in this sample. So instead of only amplifying and sequencing one type, for example, DNA, you are amplifying everything that is in the sample, therefore allowing the sequencing of viruses, bacteria, and other microbes. So, of course, this has one main disadvantage, which is that those genomes that are less represented, such as viruses, which are usually around 1 to 2% of the sample, are underrepresented and sometimes cannot be detected due to the overamplification of other types of genomes, such as bacteria, for example. So when we got to this conclusion, my boss asked me, basically, how can this data be analyzed, and is there a limit of detection when looking at the sequencing results? So this was the main objectives of my master thesis. The first one was to implement a study of sensitivity, which I'm not going to go into because it's quite complex, and then to develop a bioinformatics pipeline for the data analysis of this kind of data. So, of course, if you remember, the master's that I was doing was based on translational biomedical research. So this pipeline that I was developing had to be applied somewhere for me to be able to propose and expose my master thesis. So these were applied to two main types of samples, the first one being mosquitoes from the Amazonia. This was interesting because if we could do metagenomics in these samples, we would be able to see if there were any potential viruses that could someday be transmitted to humans through them biting us. And finally, also in fishes from Llobregat River, these fishes are in very, very close contact to residual waters, meaning that if we saw and perform metagenomics on this, we would be able to see what viruses or bacteria are in the population. So these samples were prepared through this random priming technique that I just told you about. And they were sequenced with this machine that it's called the Illumina NextSeq 2000 platform, which maybe Aina will tell you more about. And finally, I applied my bioinformatics pipeline to perform the data analysis in basically order to classify these sequences into the different types of viruses, bacteria, and everything that this sample contained. As for the results, as you can see in these graphs, for fishes, I was able to classify the sequences in different types of, in this case, domains, and also families of viruses, and the same for these mosquitoes. Specifically for viruses, I was able to find families of viruses that had been seen to be related to encephalitis or hemorrhagic fever, which are diseases in humans. However, as you can see in gray, there are a lot of unclassified sequences. There are many theories why this happened, but maybe one of these, specifically for the mosquitoes, these mosquitoes, if you remember, these are from the Amazonia, meaning that maybe these sequences and species that are there have not been really sequenced or investigated. So that's why maybe they are not in these public databases where I'm doing this classification, and that's why they appear as not classified. So once I finished my master thesis, I was really interested in this metagenomics thing, and I decided that I wanted to pursue a PhD in the same lab that I'm in. So as for my PhD thesis, it's basically based on the further development of bioinformatics pipeline. Also, I'd like to highlight that this is not a PhD in bioinformatics. It's in biochemistry, molecular biology, and biomedicine. However, my research line is genomics, proteomics, and bioinformatics. So going back to this development of the bioinformatics pipeline, the aim is to basically apply it, among other things, to real samples and see if this actually works. So as you've seen, I've applied it to mosquitoes and fish, but I will also be applying it to bats, and also more specifically human samples. So for example, cerebrospinal fluid and plasma. But more specifically, in what I am interested about, and it's what it's my PhD thesis basically, is amniotic fluid. So there's been seen that there's women that give preterm birth, meaning that they have contractions months or weeks before their due date, and usually the cause is not known. However, there's some women that come with chorioamnionitis, which is basically the infection of the placenta and the amniotic fluid. However, what is making or provoking these infections is not known yet. So it could be bacteria or viruses. we're not sure. So the aim would be to find out which viruses or bacteria are infecting these women so that this can be prevented, or there can be some antiviral treatment to treat this. So as for what the future holds, I have no idea what I'm going to do after my PhD, but for now I'm just focusing on starting it and basically finishing. And finally, I'd like to give you some take-home messages. The first one being that you enjoy university. It's really important, and obviously you need to take advantage of the professors and what you're learning, and also the resources that you have. Also, take advantage of your curricular internship and final grade project, because it's where this will open a lot of doors for you, and you will have input from your supervisors, which will be very valuable. Also, at least in my field, there's a lot of bioinformatics-related work opportunities, so that's very good news. And finally, it's a hard degree. You need to stay focused and study hard, but if you do this, you will overcome it perfectly. Thank you for listening, and if you have any questions.
For the nice talk and for illustrating all your career, the start of your career. So, time for questions. Yeah. So, yeah, you can ask questions now, and you can ask also more general questions to both of them at the end of the session.
Hi. About your project of pregnancy complications, which were the results that you obtained?
So from my final grade project, you mean? We obtained that there were proteins that were secreted into the mother's blood that were actually altered during pregnancy, meaning that if you suspect that someone has a pregnancy complications, you could do just a blood analysis and see if these proteins are altered, therefore meaning that they have a certain pregnancy complication.
Okay, thank you.
Hi. How did you start the process of contacting and searching for labs to work for?
Well, it's a bit difficult, but I basically knew that I wanted to go to the University of Cambridge, so I started looking at their websites, and if you have like a topic that you are interested in, for example, neurology or something like that, just look at the department and look at the labs and start sending a lot of emails, and someday someone will answer you.
Hey, I had a question on the master that you did on genomics. Do you have trouble to get in or something? Because I know they have like a pre-required subjects that you have done in college, but like here we are more focused on bioinformatics and not second year and third, not as much on genetics and all that.
Yeah, I didn't have any difficulty going into it, and they are actually happy to have different profiles. So if you are, well, I'm not saying that you will get in 100%, but usually if you go to a master's that is really biology related, they will accept you because they are looking for other profiles.
Okay, thank you.
You're welcome.
Any other question? No? Okay, so yeah, thank you very much. You will come back later.
Yeah.
So second speaker is Aina Montalbán. Also, as I mentioned, same promotion, second, and she also graduated 2020. She will present the talk, Contributing to the Diagnosis of Diseases Through Clinical Bioinformatics. Thank you very much, Aina.
sonando ya. Hola, hola. Sí. Good morning, everyone. I'm Aina. Today I'm going to talk about one possible path that you have when you finish a degree, that is working in a hospital. I will divide my presentation in three main parts. So first I will give you a brief context of where I work. Then we will see what is clinical bioinformatics, so how bioinformatics is applied in a clinical environment. And then finally we will see what do we need to work in a hospital as a bioinformatician. So let's start by explaining where I work. So as you've probably seen in the title, I work in Hospital Clínic de Barcelona. But to be more specific, I work at the core laboratory of molecular biology. And what is the core molecular biology laboratory? It's a clinical laboratory that belongs to the Biomedical Diagnostic Center of Hospital Clínic. The Biomedical Diagnostic Center is an institute that tries to group all the clinical laboratories from the hospital. So the main idea of the core molecular biology is to provide service to different departments. That could be, for example, molecular and genetic genetics, immunology, microbiology, pathological anatomy, with the main idea to centralize the molecular biology techniques that this department requires. And yes, so to summarize what I've just said, this laboratory where I work is a clinical laboratory, okay, that receives samples as an input. And these samples could be blood, tuber tissues. These samples belong to patients. And the objective of the laboratory is of the lab is to process the samples and analyze these samples with the final aim to that the disease specialist makes a final diagnosis to the patient. And to give you some examples about what kind of diagnosis, we can find, for example, the identification of COVID variants that we all know of a patient. Will the patient have Omicron or Alpha? This is an example. Another example could be identifying genetic alterations associated with diseases, for example, monogenic diabetes. Another example could be identifying tumoral alterations to decide which will be the best treatment for the patient. And another example is study the compatibility between donors and receptors for organ transplantations. So the lab performs several molecular biology techniques, but we will focus here on next-generation sequencing. These are the main molecular technology that the bioinformaticians are involved with, because we are responsible for analyzing this data. And these examples that you have seen here are based on next-generation sequencing data. So let's understand what is next-generation sequencing. So to do that, let's go to the beginning. There's a patient and with a diagnostic suspicion, and the clinician, the doctor, asks for a genetic test, and we extract the blood. The blood is extracted from the patient, and this sample is received in the lab. So now it starts a series of laboratory procedures that starts with extracting the DNA from this sample, or the RNA, depending on what we want to study. And we want to prepare this sample to sequence it. And how we do it, basically, we break the DNA into pieces. And it's important to say here that we don't work sample by sample. Instead, we work as a group of samples, because it's not that expensive if we work with a group of samples. And we prepare the sample for the sequencing and the sequencing start. Just to briefly mention, what is sequencing? So we're trying to read the DNA, and we will obtain the reads. I hope that you know what are the reads. This is small. We're reading these short fragments of DNA, this is the nucleotide of the DNA, and we get these reads in a file format that's called FASTQ files. And when I say we get these files, it's data, and here it starts the bioinformatician's work. Okay, so let's see how bioinformatics is applied in a clinical environment. So we have the sequence has finished, and we have some data. This data is the FASTQ files. And the FASTQ files, as you can see here, an example, is we have a read per line, and it's a very big file. It could be, for example, of 10 gigabytes. So we have millions of reads. And from here, we can do different analysis. I will explain the main one that we do, that is trying to detect genetic alterations. But as you've seen in the diagnosis, we can study the COVID-19. So there's a lot of variety, but I will focus now on this one, that is the detection of genetic alterations. So we got the FASTQ files, and from the FASTQ files, we need to convert into the BAM files. I will explain now what are the BAM files. And then once we have the BAM files, we will need to convert them into the BCF files. These, as you can see, we have a large volume of data. Okay, so I will go one by one now. So we have the FASTQ file, we have the set of reads, and we want to align our read into the reference, in this case, a human reference genome. Okay, and we map into we find the position of the read in our genome, and we got the BAM file. Okay, so for example, this read is in chromosome 22. And this one in chromosome 2. So once we have and we know the position of our reads, we can detect the variants. We can detect the genetic alterations. So for example, here in the reference genome, there's a T, but our patient has a C in this position. So here we are detecting a genetic alteration. And this makes me explain the distinctive features that the clinical bioinformatics have, that is quality. We have in this step, here, in all these steps here, in this pipeline, let's say, we need to have quality control checkpoints in order to guarantee that what we are reporting is correct, because in the final point, there's a patient waiting for her or his diagnosis. Moreover, in clinical bioinformatics, we are submitted to external quality controls to guarantee that what we do is correct and that the patient will have the correct diagnosis. Another important feature is volume. As you've seen, we have a lot of files, and we process a lot of samples per week, so we need to find ways to store this data efficiently. Moreover, we have a high diversity of analyses depending on the diagnosis, so we will need to create multiple pipelines. And another important characteristic is that in a clinical environment, the clinical bioinformaticians is fast-paced. When I say fast-paced, I mean that there's a patient that is waiting for its diagnosis, so we have the response time should be short. And in this sense, we need to automatize as much as we can of the tasks that we perform. And having said all these features, I have written what could be the day-to-day work of a clinical bioinformatician. And the main task will be analyzing the sequence samples that are received in the lab. Then another important thing is improving these pipelines, improving the bioinformatic pipelines. For example, adding more quality controls to guarantee that what we analyze and what we report has a good quality. Another thing is create, once we have a new diagnosis, or once we have diagnosis that we want to study, we can create new bioinformatics pipelines. And then another example is to generate scripts to automatize the tasks. So now that you've seen, I hope, and that you have a general idea of clinical bioinformatics, what do we need? So we need knowledge of genetics and human diseases, programming, Python, I think that you're learning now Python, R. So this is really important. I work with these tools every day. And then also concepts of databases, SQL, and why I can do why can I work here in the hospital? It's because I have a really good foundations that the degree in bioinformatics has provided me. Here, for example, I have written which are the subjects that I could say that I'm using right now in my work: genetics and genomics, all the programming subjects, omics techniques, computational genomics, data visualization. And here I've written the year that I did this subject. And another important thing that has a master, as Marta have said, is the internship. It's a really good opportunity to be able to do an internship. In my case, I did the internship, as Arnau said, in Institut Josep Carreras contra la Leucemia. Apart from the degree, I think it's good to complement the degree. And in my case, I'm doing the master's degree in innovation research in informatics, that's commonly called data science, at UPC. It's hard, let me tell you that. I've divided, it has two main topics. That's data management and all the concepts of databases, big data, and then the other concept that we study is data mining. For example, machine learning. I have some subjects of machine learning, statistical modeling, and right now I'm doing the master thesis of the master in the clinic. So even though we have studied, we learn new things every day. So in my work, I need to learn different tools and apply different tools. So here it's some of them, there's a lot. But the thing is that we are always learning every day. And just to briefly finish, working in a hospital as a bioinformatician is possible. And bioinformaticians will be and are an essential piece of hospitals for the diagnosis of diseases that are based on next-generation sequencing. And the bachelor's degree in bioinformatics will provide a very solid background, solid foundations that will, for your professional career, and you will be able to work in a hospital. And that's all. Thank you.
Okay, thank you very much, Aina. Nice that you are both pointing the similar subjects as those that you are using, that you found more useful for your research now. Okay, time for questions.
When you perform an analysis from samples, which type of professional does the final interpretation?
It is mainly done by genetists and some biologists as well. But the genetists that are specialized in a disease, is the ones that they perform the final interpretation of the results.
Okay, thank you. And I have another question, which is more general. And what weighs more in a bioinformatician, the programming skills or the biological background?
I cannot answer that because both of them are important. Both of them.
Thank you.
It's
Hi. Where do the bioinformatician work end?
okay. That's a great question. It ends when we report the files, the variant files, the BCF files that you've seen here. So we don't do the interpretation. We say, here you have the data, and now you have to interpret this data.
And there are like the possibility that a bioinformatician does this work and then can help in the diagnosis, like you have this.
Yeah, I think the possibility exists. I think the way, for example, clinic works, it doesn't fit a lot, but for sure you can do both things.
Hi. I know that sequences are different with what you work with, but how much does it usually take since you take a sample to when it's fully analyzed with all the steps you mentioned before?
If there are no problems, because sometimes there's problems, let's say 7 days. There's 3-4 days of laboratory, and then sequencing could be 2 days. So, and then we apply the bioinformatics pipeline. 1 day more, 7 days more or less. Depends on the sample and the diagnosis, the final diagnosis.
Thank you.
Do you think that when you leave the degree, you have enough knowledge to get directly to work?
It depends. Well, it depends on the internship that you're doing as well, because it's your first time where you can apply everything you've learned to something real, and I think you can kind of see how's your level, and then from then decide.
Yeah, the internship will help you to think that you can work when you finish.
Hi. What is the process to follow when the work that you've done doesn't pass a quality control check?
So the analysis stops there, and you cannot continue because the patient cannot have a diagnosis. With this sample that we are processing, we cannot continue the process because it doesn't have a good quality. So we have to stop there and repeat again the process.
Any other question? Remember, this is your last chance, and it's mandatory that you have asked a question in one of these talks.
The quality control, what is exactly like?
Okay, so first, once the sample is sequenced, you have to check that the sequencing has gone good. Okay, so we have a good quality of the sequencing. This means that we have read the DNA correctly. Then, this is the first check. Is the sequencing has gone well? Okay, then we process these files, and when we process these files, we have to check that there's, how can I explain this? We have to check that the alignment to the reads is correct, or at least it has a good quality. And then the third thing is the clinical validation of this, of the sample.
Okay, so I don't know, final question? No? If not, let's thank Marta and Aina again. Okay, so that's over. So if you want to ask, I don't know, questions in person, just come here.